Hi there. So I'm going to give you a short presentation explaining some of the work that I've carried out here at the Hawkesbury Institute for the Environment at Western Sydney University as part of my PhD. And I'm going to be talking to you about my talk entitled Siliceous, Saviour of Sugarcane, looking at the impacts of silicon on below-ground herbivory under elevated atmospheric CO2. So, as I'm sure many of you are already aware, our global climate is changing. And prior to the Industrial Revolution, atmospheric CO2 concentrations were stable for the previous 1,000 years. But now we're reaching concentrations of about 400 parts per million, and these concentrations are expected to be at least double what they were prior to the Industrial Revolution by the end of this century. And so these changes in our atmosphere are obviously going to be impacting on plants, and this is then also going to be impacting on crop production. And so the changes in the atmosphere is going to also be fundamentally altering the relationship that plants have with herbivorous insects. And that is something that I am interested in. But firstly, what are some of the general responses that we see from plants to elevated carbon dioxide? Well, one of those responses that we do see is an increase in the carbon to nitrogen ratio. So plant material typically has a much higher carbon to nitrogen ratio compared to that of insects, and therefore insects have a dietary limitation to nitrogen. And so when we see an increase in the CN ratio, we're actually diluting that, we're seeing a dilution of the nitrogen even further, and this is thereby lowering the quality of plant food as a food material for the insects. Interestingly, there's actually no general phytochemical response to elevated CO2. However, there are one class of defenses that we do see a trend, and that is in the phenolic-based defenses, which typically seem to increase in response to elevated carbon dioxide. But plants don't only rely on carbon-based defenses to defend themselves against pathogens and herbivores. They also rely on silicon-based defenses. Now, silicon has been known for many years to increase the growth and yield of many plants, but not all plants take up silicon to the same extent. They're either classified as passive, even restrictive, or active accumulators of silicon. And a recent study found that more than 10% of angiosperm families were classified as active silicon accumulating plants. But more interestingly, perhaps, the same study found that almost 58% of angiosperm families had no data regarding their silicon concentrations, which shows just how much work still needs to be done in this area. But generally speaking, the grasses are very high silicon accumulating plants. Rice, for example, can be more than 10% dry weight of silicon. And aside from the increases in growth and yield, we also see that silicon induces activity of plant defense enzymes and increases inducible defenses as well, so phytoalexins and herbivore-induced plant volatiles. Now, plants take up silicon in the form of monosilicic acid, which naturally polymerizes in the plant tissue when it reaches certain concentrations, and it forms things called phytoliths, also known as silica bodies. And these are just some scanning electron micrograph images of phytoliths in plant material taken by Professor Sue Hartley's group. And I don't know about you, but I certainly wouldn't want to be munching on any of those. They do not look particularly palatable or digestible. And that's actually one of the main mechanisms through which the phytoliths work. They reduce the overall digestibility of the plant material, and this is thought to be achieved through mechanical protection of the parenchyma cells, where insects retrieve most of their starch and protein. In addition to this, there's also good evidence of mandibular wear, so the feeding mouthparts of the insects wear down as they're feeding on high silicon diets. But there's also emerging evidence that silicon plays more than just a role in the feeding behavior of insects. Interestingly, there's an emerging theory that silicon-rich species are able to partially substitute carbon for silicon, thereby allowing them to reallocate their carbon elsewhere more effectively and more efficiently. And indeed, several studies have linked the radiation of the grasses during the Oligocene-Miocene epoch to declining atmospheric CO2 concentrations. And so at this time, CO2 at certain points was very low, and plants were therefore able to fix less carbon. And so many plants were then switching to silicon for structural function over carbon. And at the same time, we actually saw the appearance of large mammalian herbivores that were adapted to feeding on high silicon plant material. And this suggests a possible direct plant silicon herbivore co-evolutionary arms race. And so if we've seen such strong responses to changes in our atmospheric CO2 in the past, possibly then we're seeing changes in the future. So I'm then interested in how will elevated CO2 impact herbivores, and what role will silicon-based defences play in the future. So I wanted to test this. So I went about testing this looking at sugarcane and the cane grub. So sugarcane is grown in several places around the world, but it is grown in Queensland and also in far north New South Wales. Now the cane grub, that is the larvae of the greyback cane beetle, which is a native insect to Australia. And these larvae live below ground and they feed on the roots of grasses, but are typically known as very destructive feeders of the roots of sugarcane. And in fact, they cause losses of up to 40 million Australian dollars annually. So I then wanted to look at this. So how did I go about testing the impacts of elevated CO2 and silicon? I carried out a fully factorial glasshouse experiment, and my treatments were ambient and elevated CO2 and minus and plus silicon. So half the plants were supplemented with silicon in the form of sodium silicate. And throughout the growth period, I took several growth measurements as well. And towards the end, in the last 3 weeks of their growth, I applied cane grubs as well. So half of the pots were given cane grubs to feed on the roots, and these were taken out upon harvest. So on harvest, I separated the plants and dried the material for analysis. And I also took fresh root material as well to carry out in-depth feeding trials to work out certain food utilization efficiency outputs such as relative growth rate and consumption. So let's have a look at some of the responses that I got, firstly from sugarcane. So just looking firstly at the overall biomass, we can see that the grey bars are ambient CO2, the green-blue bars are elevated CO2, and you can see under the silicon minus treatment, the elevated CO2 is significantly increasing the biomass. And in fact, we see the same response under the silicon treatment as well. So under silicon, we're seeing an increase in biomass and also under elevated CO2. And this image here is just looking at 2 sugarcane plants. And this is just after a couple of weeks of growth. And the one on the right-hand side is supplemented with silicon, while the one on the left is not. And you can clearly see a difference even at this early stage of their growth. So from this, we can take home that silicon and elevated CO2 increase overall biomass. I also measured the silicon concentration in the plant material. So this is looking at the leaves and the white bars and also the roots. And you can see that under the silicon treatment, we had a significant increase in both the leaves and the roots in response to this, basically confirming that the silicon treatments were being taken up and accumulating in the plant material. Now, if you remember, I also mentioned the carbon to nitrogen ratio and how this is expected to increase in many plants in response to elevated CO2. So I measured the carbon and nitrogen in the sugarcane, and I did find a small but significant increase in the CN ratio of the roots. And interestingly, I actually found the opposite. I actually found a decrease in the CN ratio of the leaves, which could potentially indicate that the leaves are increasing their nutritional value under elevated CO2. But that's something that would need to be followed up on in the future. But we're definitely seeing an increase in that CN ratio in the roots. So silicon increases silicon content, silicon treatment, while elevated CO2 increases the root CN while decreasing the leaf carbon to nitrogen ratio. Now, if you remember what I mentioned earlier, that many high silicon accumulating plants can partially substitute carbon for silicon for structure and also for defenses. Well, I then analysed my own data to see if I could find a similar relationship, and indeed I found a strong negative correlation between the overall carbon concentrations and silicon concentrations in the plants. And I also looked at carbon-based defense to see if there was a response there. So I looked at the phenolics, and I actually did find in both the leaves and the roots a significant decrease under the silicon treatment of total phenolics. So this is therefore possibly more evidence that there is a silicon carbon negative relationship in high silicon accumulating plants. as we definitely saw under our silicon treatment, a reduction in the overall phenolic-based compounds. So, but what are the cane grubs doing in response to our treatments? Well, these two plots here are showing the mean change in grub mass. So that's the change in the mass of the insects over a certain period. So the plot on the left-hand side there is a long-term feeding experiment. So that was when the grubs were placed in the pots for 3 weeks. And you can see here again, the grey bars are ambient CO2 and the green bars are elevated CO2. And under elevated CO2, we're seeing a significant increase in the change in mass. So they're gaining more weight under elevated CO2. And this is also reflected in the plot on the right. So from the short-term feeding trial as well, we can see they're also gaining more weight under elevated CO2. But with the application of silicon, we can see a significant decrease in their change in mass. So the plot on the left, we can see they're gaining much less mass when under high silicon diet. And then the one on the right, the plot on the right, the grubs basically just lost weight when they were feeding on plants grown under high silicon. So we can see from this that elevated CO2 increases the grub mass, while silicon decreases this. So these are two further plots looking at the cane grub responses. The one on the left is looking at the relative consumption, so the amount of root material consumed, and the one on the right is the relative growth rate. And you can see in the relative consumption that under elevated CO2, the grubs are consuming more root material, and this is reflected also in the growth rate, interestingly. So we see that under elevated CO2, their growth rate is actually elevated. But when under the application of silicon, we can see that this brings it right down. Looking at the relative consumption, it's brought the consumption right down. There's almost basically no difference between the ambient and elevated CO2 anymore. And again, in the relative growth rate on the right, you can see it's brought their growth rate right down into the negative. So again, they were just losing weight basically. So elevated CO2 can possibly increase the root consumption and it can increase the growth rate. But silicon dramatically brings this back down. It really decreases their consumption and growth rate. So that's a lot of information I've given you, but what are the main take-home messages from this? Well, we can see that silicon and elevated CO2 increase the growth of sugarcane. We can see that elevated CO2 reduces the root nutritional value, but increases the plant nutritional above ground value. We're seeing evidence of that silicon-carbon negative relationship, where we're seeing under high silicon, we're seeing lower concentrations of just elemental carbon, we're seeing lower concentrations of carbon-based defense, the total phenolics. We're also seeing that under elevated CO2, cane grub growth is increased, and the performance and the consumption is also increased. But the application of silicon dramatically decreases cane grub performance and also dramatically reduces their consumption rate of the root material. So from this, we can take that silicon defenses should be a focus for the future of food security. So in the future, as we're expecting atmospheric CO2 concentrations to continue to increase, we need to look for alternative, novel strategies of pest control. And silicon-based defenses is a possible avenue for us to develop environmentally sustainable, but also extremely effective control strategies against insect pests. I would just like to thank Sugar Research Australia, who funded my research here, and also Hawkesbury Institute for the Environment, Western Sydney University. And also I'd like to thank just a couple of people who helped me carry out the research. Thank you.